plasma membrane. Plasma membrane is otherwise called as a cell membrane also, otherwise plasma lemma. Plasma membrane is a living membrane which is present in almost all the living cells. So, where it is present both in plants and also in the animals. So, here this membrane is lipoproteinaceous, that is made up of lipids and proteins, but the composition of lipids and proteins will be differing. So, here if you observe the major composition, the lipids are of 20 to 79%, whereas proteins are of 20 to 70%, and carbohydrates are from 1 to 2%. These are the major components which are present in this one. Then if we take the human RBC, the proteins are of 52%, whereas lipids are of 40%. Whereas in the myelinated neuron, where the proteins are of only 20% and lipids are of 80%. Likewise, we can see difference in the composition of chemicals which are present in the plasma membrane, but basically they are made up of lipids and the proteins. Apart from the lipids, proteins and carbohydrates, they also contains some amount of water in them, that is of nearly 20% in its composition. So, likewise this is a basic chemical composition present in the plasma membrane, and this is a structure. Plasma membrane is a living membrane, as we said. So, here it is closely oppressed on inner side of cell wall. Actually, if a cell is in turgid condition, turgid means if the volume of the cell is reaching to the maximum level. So, in such cells we can't observe the presence of plasma membrane because it is closely attached to inner side of the cell wall. But when does we observe this structure means in a plasmolysed cell. Plasmolysed cell means when the cell when endosmosis will occur, the water will go out of the cell. At that time what happens, the cell volume will decrease, or the cell cytoplasmic contents will contract. At that time the plasma membrane is separated from the cell wall. So, in such plasmolysed cell, the plasma membrane is clearly seen. Then this plasma membrane is considered to be the outermost living limit of a cell, because of course in a plant cell, apart from the plasma membrane, the cell wall is present, but as cell wall is a non-living membrane, so outermost living limit will be the plasma membrane. So, here what are the type of carbohydrates which are present in this plasma membrane means generally these are oligosaccharides. Either these oligosaccharides may be branched or unbranched structures. Then like glucose, fructose, such type of oligosaccharides are present in the carbohydrate composition of the plasma membrane. And if it comes to the lipids, phospholipids and phosphoglycerides may be present in this one. So, this type of composition will be there, and based on the nature, so the elastic nature will be there as we have said, but one more thing that this is selectively permeable and also semi permeable. It is selectively permeable to the solutes, whereas it is semi permeable to the water molecules. it will not allow all types of substances to pass through it. So they have some the plasma membrane will contain some specialized membrane structures which are helping in selecting some particular type of ions to pass through them. So it will not allow all types of ions or all types of the substances to pass through. So that type of ability is there in the plasma membrane called as selectively permeable nature. Many models have been proposed by various group of scientists to explain the structure of this plasma membrane, and before that, so the scientist who first discovered this plasma membrane is a Schwann scientist. So because the Schwann is a German zoologist who first said that the animal cell does not have the cell wall, but it is surrounded by a living membrane. It is surrounded by a membrane called as a plasma membrane. Then coming to the various models which have been proposed to explain the structure of the plasma membrane, in that the first one is a sandwich model. This model was proposed by two scientists, one is Danieli and Davson. Actually here the proteins and lipids are present, as we said. So lipids have one special feature. The lipids have both hydrophilic nature and the hydrophobic nature. So then they contains the hydrophilic or polar head, which is present towards outer side, whereas they contain hydrophobic or non-polar tails, so which are fatty structures which are present towards inner side. So this type of nature is there with the lipids. So hydrophilic polar heads are present and hydrophobic non-polar tails are present. And here if you could see the diagram, so this is a head region where tails are present, which are present towards the center, and according to this model we can see the two layers of lipids. Likewise, so we consider this as a bilayer of lipids, and this bilayer of lipids are undifferentiable, that is they are not separated, they are present just one after the other like this. So that's why we call it as a bilayer of lipids. And on outer side we can see the globular protein molecules. This forms a globular protein molecules which are in the form of a layer. On other side also we can see the presence of globular proteins in the form of a layer. So here this is a phospholipid bilayer or two layers, and on outer side this is one more globular protein layer, and on other side also we can see one more globular protein layer. So actually they consider that there are 4 layers are there, 1, 2, 3 and the 4 layers. But as these 2 layers, the 2 phospholipid layers are not separated from each other. So we consider this as one single unit, second layer and the third. Likewise the differentiation can also be seen. So here this part is a hydrophilic head region and this part is a hydrophobic tail region. The hydrophilic head of the phospholipid layer and the globular proteins are held with electrostatic forces, whereas the tail of one phospholipid layer and the tail of the other phospholipid layer are binded with van der Waals forces. So this type of binding is also been seen in the phospholipid and the globular protein structures according to the sandwich model that is proposed by Daniel and the Davson scientists. And here the arrangement of the phospholipids and the globular proteins in a systematic and a linear manner. So they are not being disturbed, or we can say they are present in a continuous layers. So here there is a little modification of the Daniel and Davson's model. So that was given by scientist called as Robertson. So here we call it as a unit membrane model. Unit membrane model. According to the scientists, 3 layers are present. So, trilamellar, tri means 3, lamella means layers. So, 3 layers are present and all the 3 layers are considered to be a single unit. So, we call it as a unit membrane model. And according to this Robertson scientist, here the phospholipids are present in the center. So, which is considered to be the bilayer, but as a one layer only. And on either side, as like in the Daniel and Dawson's model, the protein layers are present on both the sides of the phospholipid bilayer. And here also we can see the hydrophilic head and the hydrophobic tails. But here the proteins which are present on outer side are different from that of the one which are present in the towards the inner side. So, this here we call them as mucoproteins which are present on outer side, extrinsic one, whereas these are non-mucoid proteins which are present towards the inner side. Non-mucoid proteins towards the, this is what the cytoplasmic side, this is the external side or outer side. So, this type of change can be seen in the type of proteins which are present in the external outer side and the inner side according to Robertson. So, this type of concept given by Robertson called as unit membrane model, and according to the scientist, not only the plasma membrane but all the living membranes are having the same type of organization, that may be a plasma membrane or the membrane of the cell organelles. So, this concept where each and every living membrane in a plant cell is having the unit membrane like organization, and that he, according to the scientist, the total thickness of this membrane is of 75 angstroms. The total thickness is 75 angstroms. Out of this phospholipid bilayer is of 35 angstroms in the thickness, whereas the protein layer, this protein layer is of 20 angstroms thickness and this protein layer is of 20 angstroms thickness. So, here the total thickness comes to 75 angstroms thickness. The same type of thickness is approximately there for each and every living membrane according to the unit membrane model. Next one is fluid mosaic model. This fluid mosaic model was given by Singer and Nicholson. So, according to the scientists, so it is not a solid type of structure, it is a quasi fluid in this nature. So, where these proteins are present and the lipids are present. Here, as like in the earlier models, we said that proteins are present in one uniform layer and lipids are present in other uniform layer. But here a little difference is there. Here the lipids are present in uniform structure in the center as like that of a sandwich model or the Robertson's unit membrane model, but the proteins are present in a mosaic pattern. That is, a irregular arrangement of the proteins are present here. That is a special feature, and the fluid is the lipids are present in the uniform fluid manner. The fluid lipid layer is present, and in that fluid lipid layer many proteins are irregularly arranged in a mosaic pattern. This type of organization where the quasi fluid nature is present. This quasi fluid nature is responsible or useful for various properties of the plasma membrane like repair, growth, cell division, and also the selective permeable property. All these properties are explained because of the satisfied by because of the fluid mosaic model, and that is the reason why it is more acceptable theory when more acceptable model when compared to the other models proposed by other scientists. So, according to this one, the proteins are present. Two types of proteins are there. The one which are towards the outside are called as the extrinsic proteins, and the one which are embedded inside are called as the intrinsic proteins or integral proteins. So, here integral proteins are present along with the lipid layer. So, let us see that structure in the form of a diagrammatic representation. Here all these phospholipids are present in the bilayer, as we had seen in the earlier models also, like this. So this is a uniform arrangement of the phospholipid bilayer. Here also we can see the presence of this is a hydrophilic head, that is polar region, and this is a hydrophobic tail, that is a non-polar region. And so here and there we can see some from out upper side also we can see the spherical shaped structures, but apart from this one, here and there we can see some proteins which are embedded, which can't be seen from outside. So this type of proteins are called as the integral proteins or the intrinsic proteins, but some proteins can be seen from outside like this. So these are called as the extrinsic proteins. So this head-like structures can be seen. This is nothing but the hydrophobic head regions of the proteins, hydrophobic head regions of the lipids. And here and there we can see the extrinsic proteins like this, the one which can see from outside. So these are embedded, which we can't see from outside. These are called as a intrinsic proteins. And apart from this one, here and there we can also see some carbohydrates in the form of oligosaccharides like this. And these oligosaccharides may be either the branched or unbranched structures, which are nothing but the carbohydrates, and sometimes the glycoproteins and glycolipids can also be seen here and there, which are small carbohydrate molecules. So we can call them as glycoproteins and glycolipids. Apart from that one, here and there we can see the little other substances like cholesterol. So various other structures have also been observed in the fluid mosaic pattern of the plasma membrane. And here let us discuss something about the intrinsic proteins, which are also acting as the channels for transportation of materials, or particularly the water from outside to inside. So here, for example, if you see that this is a phospholipid layer, bilayer. Here we can see some protein molecules like this, which can be seen towards the outer side also. This type of proteins which are acting as a channels for transportation of water, and we call such structures are called as a transmembrane proteins. These are called as a transmembrane proteins, and these transmembrane proteins are otherwise called as tunnel proteins also, because they are acting as a tunnels for transportation of the water, or we can call it as water channels also. So this type of membranes are also be there, and there are some proteins which are the integral part of the plasma membrane, concerned with transport of ions and other substance also. So those are also called as transmembrane proteins. So here these transmembrane structures are formed either by one protein or the adjacent proteins which are present in the plasma membrane. So here this type of organization which are having the transportation ability, and there are some proteins which are present in the plasma membrane which are having the enzymatic nature also. The plasma membrane will help in the transportation, as we are saying. So the movement of the substances will occur by various methods, either by using energy or without using energy. Based on that one, the movement either may be by passive method, so we call it as a active absorption, or the passive absorption, or the passive movement of the substances, where always the movement of the substances will be in the downhill movement. Downhill movement means the movement will always from the high concentration region to the low concentration region. and here energy is not utilized. So this type of movement where energy is not utilized and from high concentration to low concentration is called as a passive movement. And here sometimes the neutral substances are transported by passive diffusion, where the energy is not utilized. The same characters downhill movement or from high concentration to the low concentration region. The lipid soluble substances are also being diffused by the same, transported by the same passive diffusion movement. And there is one more passive movement is called as facilitated diffusion. So in the facilitated diffusion also energy is not utilized, but here the specific type of ions are showing the mobility from one side to the other side. Then apart from this one, there is one more method is that is called as a active diffusion or the active movement or active transportation, where this is the process where always the movement of the substances will be from in the uphill direction or uphill movement we can say, or otherwise from low concentration region to high concentration region the molecules will be moving. And for this process they require energy. So energy is utilized, and energy in the form of ATP is utilized for this process. So here the movement is from low to high concentration we are saying. So for this process they require some membranes which are present in the plasma membrane. Those membranes are called as a transmembrane proteins. These transmembrane proteins have some specific sites on them. So those specific sites, this ions or the substances which have to be transported will go and bind to that specific sites, and after that by using this ATP energy, so there some conformational change or the structural change will take place in the protein membranes, so that they will be showing some sort of change, so that the substance will be exposed to the inner side of the cell where they are releasing that substance. This type of change where the energy is helping in the conformational change is called as a active transport of the or active movement of the substances. In the active movement, here there are some mechanisms where sodium and potassium pump is present, or we can sodium pump and sodium ion potassium ion pump, or the ATPase sodium H plus ATPase mechanism is also there, where the energy is utilized for transportation of the ions from one side to the other side. Then apart from this one, here the movement of ions will be taking place through some specific channels. We call them as the water channels or the aquaporins, through which the movement of water will be taking place. Then apart from that one, based on the movement, so we call them as uniporters or uniport mechanism. Uniport or uniporters means if a ion is if a transmembrane protein is allowing the movement of one ion from one side to other side or in one direction, that is called as uniporters, and the mechanism is called as uniport, and other is there called as co-transport. Co-transport means two types of ions are moving at a time. This type of when two ions are moving at a time we call this co-transport. In this also antiport and symport are present. Antiport means the movement of the two ions in opposite direction. One ion is moving from inside to outside and other ion is moving from outside to inside. Whereas in symport, both the ions are moving in the same direction simultaneously from inside to outside, or both the ions are moving simultaneously at a time from outside to inside. So this type of coordinating movement of two ions is called as in the same direction is called as symport, and the two ions moving in opposite direction at the same time is called as antiport. These two are the part of co-transport. Likewise, the transmembrane structures are helping in the movement of the ions in one direction or the two directions, or at a time one ion or two ions. Based on that one, uniport and co-transporters are present. So all this mechanism of co-transport and uniporters are useful for the active transportation of the ions from one side to the other side of the plasma membrane. Inside the plasma membrane, the protoplasmic contents are present. We call it as a protoplasm, or already we said the protoplasm is the one which contains cytoplasm and the nucleus. And cytoplasm contains cytoplasmic matrix, so that otherwise we call it hyaloplasm, and in that cell organelles and the other inorganic and organic components are present. So here in that one, let's discuss about the cell organelles. So in the cell organelles, some will be having the functions which are with the coordination with each other. Such type of structures are called as a endomembrane system. There are some cell organelles which are closely coordinating their functions, having close coordination of the functions. Such type of structures are combinedly called as endomembrane system, which includes endoplasmic reticulum, Golgi complex, vacuole and lysosomes. These four are combinedly called as endomembrane system. And why not other cell organelles are not forming the part of endomembrane system is the functions of those other cell organs are not in close coordination with each other. So that's why the other cell organs are not the part of endomembrane system. So let's discuss some other cell organelles. First, the cell organs which are the part of endomembrane system, starting with endoplasmic reticulum.